Welcome students to today's YouTube live class. This is today's botany class, and the topic for today is the viruses and viroids. So what are viruses and viroids? We shall discuss today. Before, in the previous classes, we had discussed up to kingdom fungi of the 5 kingdom classification system. So in this 5 kingdom classification system, the position of virus has not been ascertained. So the viruses have not been given any position in the 5 kingdom classification system. So the viruses, what are viruses? We shall discuss today and how they replicate, because in today's time we come across or we hear about lots of viral diseases that are now prevalent, and what are actually these viruses and how they replicate inside a cell. So virus and the viroids are acellular particles, that is, they do not have a cell structure. So that is why they have not been assigned any position in the 5 kingdom system. So what are actually viruses? If we define the virus, a virus is an acellular, without a cell structure, non-cytoplasmic particle, and this has only the nucleoprotein and the genome as its constituent and does not have any cytoplasm. So the basic activities that occur inside a cell does not occur in the case of a virus particle. We shall discuss what is the discovery of the virus. So the topic today we shall discuss is the viruses. So how viruses were discovered? What is the discovery of virus? So before the viruses were discovered, it was always known that the bacteria are the smallest living organisms or the pathogens that cause disease. So in 1892, the Russian botanist Dmitry Ivanovsky is credited with the discovery of the virus. So he was working on the tobacco plant and he noticed a disease where the tobacco leaves had a mosaic symptom. So mosaic is white patches that were present on the leaf, and he was trying to find out what was the causal agent of this disease in tobacco plants. So when he tried to isolate the bacteria or the causal agent, he could not isolate any such structure, but he found that when the extract of the tobacco leaves which are diseased were taken and they were sprinkled over the healthy leaves, again the healthy leaves also started producing the same symptoms or the disease occurred. He tried to isolate the organism thinking it was a bacteria through a bacteriological filter, but he could not get any bacterial structure on the filter. So he said there is some structure in the fluid that is actually causing the disease. So in 1898, Bejernik, he coined the term virus. So the term virus was coined by Bejernik, and this term virus, in the Latin term, it is the word vira, which means poison or venom. So it was named as poison and venom, and so he said it is contagium fluidum, that is, it is a fluid which is contagious or which is spreading the disease, and the name vira was given as poison or venom because no structural particle could be isolated. So in 1935, for the first time, W.M. Stanley, he was able to isolate the virus particles, crystallize the virus particles, and he said the viruses are particles made up of nucleoproteins. So viruses are particles made up of nucleoproteins, that is, they contain a nucleic acid component, DNA or RNA, and a protein component that forms the particle of the virus. So he was also awarded the Nobel Prize for finding out the fundamental structure of the virus particle in 1946. So, this was the basic discovery, and the first therefore discovered virus was the tobacco mosaic virus, or commonly called as the TMV virus. So, we shall see the discovery in a slide. So, the discovery of the virus, the main points of discovery of virus is that viruses are acellular, non-cytoplasmic infectious agents, and Ivanovsky is credited with the discovery of the virus from tobacco plant extract, and Bejernik called it as Contagium vivium fluidum and coined the term virus, which means venom or poison. And W.M. Stanley showed that viruses could be crystallized and they are composed of nucleoproteins. So, we shall discuss now the general characters of viruses. What are the general characteristic features of the virus particle? So, the virus particle is the acellular structure. They are non-cytoplasmic. They do not contain any cytoplasmic structure or a cell structure, but they are always infectious. So, these viruses cannot multiply on their own, and they are obligate parasites. So, in the second point you can see they are obligate parasites and they multiply only inside a living host cell. Obligate parasite means they are always a strict parasite. They cannot have any other form of existence. They will be always parasites and multiply only inside a living host cell. So, a living cell is required for their multiplication, and they will multiply themselves only if there is a living cell. It can be a plant cell, animal cell, or a bacterial cell. So, the composition of the virus, what it is made up of? The viruses are composed of nucleoproteins, and this nucleoprotein is two component. One is nucleic acid and the other is the protein component. So, the nucleic acid forms the viral genome or the genetic matter of the virus particle. This virus genome can be either DNA or RNA, which can be of the double stranded type or of the single stranded type. In the virus particle, DNA and RNA are never found together. So, the viruses are always much smaller than the bacteria, and they can pass through the bacteriological filters. So, we cannot hold the viruses on the bacteriological filter because they are much smaller. When we measure the bacteria size, normally we measure them as micrometers, whereas the virus particles are measured as nanometers. So, here they are always host specific, that is a particular type of virus has some host that is specific for it and can multiply only inside that host cell. And these viruses can be easily transmitted from one host to another. Like for example, the COVID virus being transmitted from some animal to the human beings, or that is the possibility that we see, that is the bat can be a transmitter to the human host. So, it can be transmitted from one host to another, and these viruses can be cultured in the laboratory on only living cells. So, always a living cell is required for the existence of a virus particle. So, the next is the size and shape of the viruses. The size varies between 10 nanometer to up to 280 or 300 nanometers. So, 1 nanometer is 10 to the power minus 9 meter. The size will vary between 10 nanometers, which is the smallest virus, as in case of foot and mouth disease virus of cattle, to the variola virus or the smallpox virus, which can be up to 280 nanometers. So, the shape of the virus can also vary. we can find different shapes of the viruses. The shapes of the viruses can be complex, it can be helical, or it can also be icosahedral. So, you can find them as golf ball like structure, you can find them in the shape of a rod shape, a polyhedral shape or a cubical shape, or the complex is the tadpole shape in case of the bacteriophage that is seen in the figure. So, these are different shapes of the virus. So, we shall now discuss of the general characteristic or general structure of the viral components. So, the general structure of the viral components can be divided into two parts. So, if we take a viral particle, the virus particle can be divided into two different components. As said earlier, the viruses are particles of nucleoproteins. So, the virus particle, if we take the simple structure of a virus particle, the virus particle will have two components. One is a protein cover that is present, and inside this protein cover you will have the genetic material tightly packed. So, this component will be the nucleic acid component and this will be the protein component. So, what is the characteristic feature and importance of these two components? The protein component of the virus particle is called as the capsid. The protein component of the virus particle is called as the capsid. So, this is the protein component. So, these protein molecules are made up of different types of amino acids to form specific proteins which form the capsid. So, in general we can say the capsid is the outer cover of the virus particle. So, this capsid in turn is made up of subunits, protein subunits. So, these protein subunits are called as the capsomeres. The protein subunits are called as the capsomere. In a particular virus particle, the protein capsid has definite number. So, for each virus particle, there is definite number of capsomere that is present or forming the capsid, and these capsomeres are arranged in a specific manner to provide the shape to the virus particle. So, the arrangement of the capsomere in the capsid gives you the shape of the virus particle. So, this protein capsid, which is your protein component or the capsid, is composed of specific number of subunits which are identical and called as a capsomere. So, the capsomere number is always definite. They are arranged in a definite manner to give or provide shape to the virus particle. So, what is the importance of this protein capsid? So, the protein capsid gives the property of host specificity. So, whenever a virus has to attach or enter any cell, the virus recognizes the host cell by its capsid. So, the capsid protein is important to actually give the host specificity to the virus particle. So, the virus particle, the first part is the capsid. In some of the virus particles, the capsid is further again covered by another extra layer, and this extra layer is called as envelope. So, if whenever the capsid is covered by another extra layer, this extra layer will be called as the envelope. The envelope may be made up of lipoprotein. So, it will be composed of lipoprotein and can have additional structure. So, whenever a virus particle is covered by an envelope, such a virus particle is called as enveloped virus, and if the envelope is absent, such a virus particle will be called as a naked virus. So, if this part is not there, then it will be a naked virus. If there is an envelope, it will be called as the enveloped virus. So, example, HIV virus is an enveloped virus and it has different spikes on its surface which help in attachment and they give specific character. So, the tobacco mosaic virus is non-enveloped or the naked virus. So, the second component, we will move to the second component of the virus particle. The second component is the nucleic acid component. So, the nucleic acid component is the viral genome or the genetic matter that is packed inside the capsid. So, in case of virus particles, there is no cytoplasmic presence or there is no cytoplasm inside the protein capsid. Only the genome is packed. This nucleic acid can be either DNA or it can be RNA. The nucleic acid can be DNA or RNA. Further, the DNA can be double stranded or single stranded. What we mean by double strand? This is normal double helix structure of the DNA where two polynucleotide chains are present, and this is single strand. And even the RNA can also be double stranded or single stranded. Normally, in the prokaryotic and eukaryotic cell, we have double stranded DNA, single stranded RNA. Whereas in virus particles, you can have either double stranded DNA or RNA, even single stranded DNA or RNA. So, what is the importance of this nucleic acid in the virus particle? The nucleic acid gives you the replicative power or infective power. So, this nucleic acid helps in the multiplication of the virus particle inside the host cell, and infection that occurs in our cell or in the body is due to the presence of the viral nucleic acid. So, the viral nucleic acid is the viral genome. So, viral genome here is either DNA or RNA. So, these two are the two basic components of a virus particle. So, we can take certain examples of the virus particle. The virus particle we can discuss two virus structures. One is tobacco mosaic virus and the other is a bacteriophage. So, we shall discuss two structures of the virus particle. So, virus particle, one is tobacco mosaic virus. The first we shall see is tobacco mosaic virus. So, tobacco mosaic virus is a rod shaped virus. So, here in the figure you can see the tobacco mosaic virus, commonly called the TMV virus, and this is the most extensively studied virus because it was the first virus to be discovered. TMV is a rod shaped virus having a diameter of about 18 nanometer and a length of about 300 nanometer. So, here we can see the protein capsid. The protein capsid here, which is seen as in the yellow color, is the protein capsid. The protein capsid is made up of identical units which are the capsomeres. So, the capsomeres here in the definite number is 2130 capsomere that is present, and these are arranged helically around a central axis. You can notice the central axis of the virus particle. So, around the central axis you can see the arrangement of the capsomere, and each of the capsomere is made up of 138 amino acids. So, amino acids present is 158 amino acids make up the capsomere. So, this capsid is about having about 130 turns in the protein helix, and each turn has about 16 capsomeres arranged. So, 16 capsomeres in one turn, and you have around 130 such turns to make up a virus particle. The viral genome in case of tobacco mosaic virus is a single stranded RNA molecule that is present in the central core arranged in a helix. So, it runs along the entire length and is present in the form of a spirally coiled helix. Some portions of the RNA, small portions of the RNA even protrudes out of the capsid and can be seen. So, there are about 7,300 nucleotides in the RNA molecule. So, this is the basic structure of the tobacco mosaic virus. Here you can see that the virus is not enveloped and a naked virus. It causes tobacco mosaic disease in the leaf of tobacco plants. So, the next is a bacteriophage. So, what is a bacteriophage? Bacteriophages are special viruses that infect only bacteria. So, the host for these phages or viruses are the bacteria. They infect different types of bacteria, and the most extensively studied bacteriophage is the T series bacteriophages, which infect the E. coli bacteria or the Escherichia coli bacteria. And the shape of the bacteriophage, the T series bacteriophage, here is called as a tadpole shape. So, the tadpole shape shows there is presence of a head and a tail. So, it is given according to the shape of the tadpole. So, the tadpole shaped virus, here bacteriophage, consists of a head and a tail. So, let us discuss the structure of this bacteriophage. Bacteriophage structure. So, the common T series bacteriophage has a hexagonal head. So, this is the head component of the bacteriophage, and then it is attached to a tail. So, the bacteriophage here will have a head and a tail showing the tadpole shape. This head, hexagonal head, is around having a length of around 950 angstroms and a width of around 650 angstroms. So, this hexagonal head will have the genetic matter tightly packed inside the head. This genetic matter here is double stranded DNA. So, this is the head component. This head remains attached to the tail by the help of a short neck. So, this is the neck part which remains attached to the head. At this tail, this is the tail core having a diameter of around 80 nanometers. Then at the base, this remains attached to another hexagonal base plate. So, the virus particles here has a hexagonal base plate attached, and the hexagonal base plate has spikes to attach to the host cell and also has the tail fibers. So, it has such tail fibers which are present. All these components here are made up of protein except the DNA component. These components are all made up of protein. Further, the entire tail core is surrounded by a contractile sheath. So, the ring of contractile sheath is present around the tail core. So, this is the contractile sheath which helps in compressing the tail part of the viral particle. This is called the contractile sheath. This contractile sheath remains attached to the neck by rings of protein which are called as the collar part. So, this is the tail fiber. These are the spikes for attachment. This is the base plate. This is the typical structure of the T series, even series phage, which is T4 bacteriophage. So, the head component here is hexagonal, packed with double-stranded DNA, which is the genetic material. The head remains attached to a tail core, and this tail core is filled with lysozyme type of enzyme, and it helps in infecting the host particles. So, there is lysozyme that is present inside the entire tail core. The tail core is surrounded by rings of protein forming the contractile sheath, helping to compress the tail during infection. Then there are tail fibers that remain, there are 6 tail fibers that remain attached to the end of the base plate. So, the base plate and the tail fiber help in attaching the bacteriophage to the surface of the host bacterial cell. The tail fibers are also made up of protein around 1300 angstroms in length. So, there are also small spikes to connect to the bacterial cell wall, and this is the basic structure of a bacteriophage. The length of this part is also approximately around 950 angstroms. So, the length of the head is also 950 angstrom. The tail also gives around a length of 950 angstrom. So, this is the basic structure of a bacteriophage. So, these bacteriophages are basically the viruses or the phages that infect different types of bacteria. They can cause disease in the bacteria and they multiply inside a bacterial cell. Let us now come to the next part of the virus, that is the life cycle of the virus. Life cycle of the virus. How does the virus actually multiply inside a host cell? The virus particle has 2 components, as we said, the nucleic acid part and the protein component. The nucleic acid part is the infective part which helps in multiplication inside a host cell. The basic steps of viral multiplication can be divided into adsorption, second is penetration, third is uncoating, fourth is biosynthesis, fifth is assembling, and sixth is the release part. So, for normally a viral particle which is trying to multiply in a host cell, these are the basic steps: adsorption, penetration, the uncoating, biosynthesis, assembly and release. So, this is the life cycle part or the replication part of a virus particle. The virus particle replication steps can be easily studied with the example of a life cycle of a bacteriophage. So, we shall discuss the life cycle of how a bacteriophage multiplies inside a bacterial cell. So, the life cycle of bacteriophages can be basically divided into 2 types. One is called the lytic cycle, and the other type of cycle is called the lysogenic cycle. The lytic cycle and the lysogenic cycle. We shall first discuss the lytic cycle part and then the lysogenic cycle part. So, let us first see the diagram of both these cycles, and then we shall I shall detailedly explain the process of the life cycle. So, here this is the life cycle of bacteriophages, showing or depicting two types of cycle. One is the lytic cycle and the other is the lysogenic cycle. So, coming to the lytic cycle, in the lytic cycle we can see how the virus or phage is attaching to the host cell, injecting its DNA, and then this DNA is now entered into the bacterial cell, then it is multiplying, and then it is coming out from the bacterial cell producing complete phage particles. So, that is the lytic cycle. Sometimes the phages do not come out of the bacterial cell, but try to multiply their DNA inside and along with the bacterial cell. So, here the lysogenic cycle comes into existence, where you can see the viral genome is multiplying or attaching itself to the bacterial DNA, and then as the bacteria multiply, then they also multiply along with the bacterial DNA. If such a cycle occurs for multiplication, then it is called the lysogenic cycle. So, let us first discuss the lytic cycle. So, the lytic cycle can be seen in case of the bacteriophage, T4 bacteriophage, whose structure we just studied. So, let us see how the lytic cycle proceeds. So, the first step of the lytic cycle, if this is a bacterial cell, this is the cell wall of the bacterial cell. The first step of the cycle is adsorption. So, the first step will be adsorption. So, what is adsorption? Adsorption is the process of attachment of the phage particle over the surface of the bacterial cell wall. So, for adsorption, the phage particle will get attached over the cell wall. So, this is the bacteriophage, the T4 bacteriophage, and this is the viral genome or the double stranded DNA. So, whenever this virus particle gets attached, so the attachment is also specific. The tail fiber particles or the proteins will recognize specific receptor site which is present on the cell wall of the bacterial cell. So, where it will attach? It will never attach randomly, but on a receptor site. So, the receptor site will be having a specific protein that can be attached to the phage protein particle. So, here the base plate, the fiber particle, with the help of the spike, they get attached over the surface or the cell wall of the bacterial cell. So, this phenomena is called as adsorption. So, here the bacteria is a living cell having also a double stranded DNA. So, this is the bacterial DNA. So, the next step, this is the cytoplasm. So, what happens after adsorption? So, after adsorption, the second step is penetration or entry into the bacterial cell. So, during penetration, the bacteriophage that is present, it will release the lysozyme that is present in the tail core to dissolve the cell wall. So, the cell wall will be dissolved for the bacterial cell. So, the cell wall of the bacterial cell is dissolved by the help of lysozyme that is released from the tail core, and the contractile sheath will compress, and then there will be the entry of the DNA of the viral particle. So, by compression of the contractile sheath, the DNA, phage DNA, this is the phage DNA, this phage DNA will now enter into the bacterial cell. So, here we have the complete bacterial DNA. So, this is the bacterial DNA and this is the phage DNA. Bacterial DNA and the phage DNA. This particle, the capsid part here, will remain outside. It will not enter into the cell. So, the phage DNA will only enter and this part will remain. The empty protein capsid that is remaining attached over the bacterial cell is also sometimes called as the ghost. So, it is now empty. The phage DNA has entered into the bacterial DNA. In the third, so this step can be written as penetration. So, this step is the penetration step. So, after penetration, what happens is the phage DNA becomes active. The phage DNA then destroys the bacterial DNA and takes over the entire machinery of the bacterial cell. So, the bacterial cell has ribosomes for protein synthesis, it has the cytoplasm, the all other requirements that is required to produce or multiply the phage DNA. So, in the next step, which will be the biosynthesis step. So, in the biosynthesis step, we find that the bacterial DNA is broken into fragments. That is, the bacterial DNA is no more active. What is active is the phage DNA, and this phage DNA takes over the entire protein synthesizing machinery of the bacterial cell. That is 70S ribosomes, RNA are all taken over by the phage DNA. The phage DNA will direct the synthesis of all proteins required for its component. The capsid proteins will also be synthesized, and then the viral components also will be synthesized. So, this phage DNA now multiplies or replicates using the enzymes present in the bacterial cytoplasm. So, this is the multiplication of the phage DNA. After the phage DNA is multiplying, then the proteins are also synthesized. This is the head part, the tail fibers, all these phage components will be synthesized inside the bacterial cell. So, this part here is biosynthesis, genome multiplication and biosynthesis. So, here now the virus is using the bacterial cell as a factory to produce its own components, and the bacterial no more, DNA no more controls the bacterial cell. So, after the biosynthesis phase, the phase will be the assembling phase, because here the parts or components are produced. After then there will be attachment. So, there will be complete production of the viral phages. So, here the phage particle will be synthesized. So, this is the synthesis of the new phage particles inside the host cell. The bacterium is the host cell. This is the complete phage particle that has now been produced. So, this is the step of the life cycle called as the assembling step, assembly or maturation. So, when complete particles have been produced inside the bacterial cell, the next step is to come out of the bacterial cell. So, the phages now will need to come out and start a new life cycle in the environment. So, here the last step here will be release, and this release step here is called as the lysis part. So, what happens during lysis? So, this is the rupturing of the cell wall of the bacterium by release of lysozyme. The lysozyme will be collected from the bacterial cell. It will be packed inside the tail core. So, the release of lysozyme again will break open the bacterial cell wall. So, when the bacterial cell wall is released or broken, the phages will be released outside. So, these are the new phages that were synthesized inside the bacterial cell. This phase here is called as the lysis. Lysis or breaking open. So, these phages will now again infect new bacterial cell and continue the cycle. So, this type of a cycle in which the phages are destroying the bacterial cell after multiplication is called the lytic cycle. So, lysis is breakdown. So, it is called as the lytic cycle. So, these T4 bacteriophages that destroy the bacterial cell after their multiplication, they are called as the virulent phages. These are called the virulent phages. So, this is the lytic cycle. The second cycle is the lysogenic cycle. So, in the lysogenic cycle, we shall discuss what happens in the lysogenic cycle. The phages that undergo lysogenic cycle do not destroy the bacterial cell after multiplication, but they multiply along with the bacterial cell genome, and they are called as the temperate phages. So, those which will show lysogenic cycle are called as the temperate phages. For example, the lambda phage, which infects the E. coli bacteria, is not a virulent phage but a temperate phage. So, what happens during this process of lysogenic cycle? The same steps continue, that is, there is adsorption. Adsorption is the attachment over the surface. So, here this is the phage and the DNA of the phage. This is the bacterial DNA. So, it will show adsorption. It will also show the next step, which is penetration. So, in penetration part, we know it will be only the viral genome that enters into the bacterial cell, and this is the bacterial genome. The steps of the lysogenic cycle change after this penetration step. So, this is penetration. So, after penetration, this genome of the virus or the viral DNA will not destroy the bacterial DNA as in the earlier cycle, but it will continue and get attached with the bacterial DNA. So, this is the viral genome which gets attached to the bacterial DNA at specific point. This attachment gives you a combination of the bacterial DNA plus the phage DNA. This condition is called as the prophage. And this bacterium is now a lysogen. So, here this is the prophage condition. In the prophage condition, you have phage DNA plus the bacterial DNA. So, both of this get attached, phage DNA and the bacterial DNA. As this bacterium then multiplies by binary fission, so the bacteria will multiply by binary fission, so also the phage DNA. So, the phage DNA also becomes 2 copies when the cell divides into 2, and this will continue multiplying along with the bacterial DNA for several generations unless it requires to come out of the bacterial DNA. If the phage DNA tries to come out of the bacterial DNA, then it converts itself into the lytic cycle and then it gets released. So, this is the life cycle, the lysogenic cycle that occurs in case of the temperate phage. So, the next condition, so here you can see the basic steps of the bacteriophage lytic cycle, which is adsorption, penetration, biosynthesis, maturation and release. So, we studied that bacteriophage which cause the lysis of the host cell are called virulent phages. The phages which incorporate their DNA into the host cell and replicate along with the host chromosome or the bacterial DNA are called as temperate phages. So, these are certain common viral diseases that are present. These are common animal disease or human disease, which is influenza, common cold, smallpox, mumps, measles, the German measles or rubella, poliomyelitis or polio, and commonly now the coronavirus or the COVID. So, the AIDS or HIV virus causing AIDS are also viruses of different types. So, the influenza virus also causes your cold symptoms. So, the common cold that we come across is also due to rhinovirus, which is a RNA virus. Similarly, viruses also cause diseases in the plants, which are little leaf of brinjal, yellow vein mosaic, and the potato leaf curl of papaya, bunchy top, grassy shoot, tobacco mosaic. These are common diseases. In the plants, mostly the viral diseases show a mosaic pattern over the leaf or a rolling pattern, and they are common plant diseases. So, the next part of this series is the viroids. So, viroids, if we discuss, they are still smaller than viruses, and these viroids are the particles of simple RNA molecules, or they do not have a capsid structure. They are having only single stranded small circular single stranded RNA molecule, which are having low molecular weight. They are without a protein coat. So, here we can see they are still subviral particles that are also present. Here the protein capsid is missing, but the RNA is capable of infection. Mostly they cause diseases only in plants and do not cause disease in animal. The famous potato famine in Ireland was also caused by such a viroid destroying the potato crops, and they resemble the introns cut out of eukaryotic gene. So, introns are small pieces of the nucleic acids, and mechanism of how viroids are actually multiplying or present is still unknown. So, these are the different viral diseases. So, viroids are sub-viral particles that are present. So, similarly, the virus particle is now having sub-viral particle which is viroids, and also there are certain particles called as the prions. So, prions here are protein molecules that are present causing disease. They cause disease even in case of the humans, and they do not contain any genetic matter. So, this is all about virus and viroids for today. Hope you enjoyed the lecture, and thank you all for patiently listening. So, in the next class we shall discuss about kingdom Plantae in detail. Thank you all.